senior research presentation you're about to see is really indicative of the kind of work that we hope that most of our students will engage in. Sustained research in an area of personal interest. What's interesting about Kevin's research is that it's a continuation of another Math and Science Academy student's work. A question that student raised at the end of his presentation for potential future research intrigued Kevin enough to explore it. And when I looked at his proposal from last year, I could tell that he'd done his homework prior to submitting his proposal. He knew about the research topic, he knew about the background and the review he'd done a review of literature prior to submitting his proposal. So what you're about to see is a great example of a senior research project. So please welcome him this morning.
Thank you, Ms. Graves. Hello, my name is Kevin Nelson, and welcome to my presentation, Reading Minds Through Brain Fingerprinting. Over the past few months, I worked under Dr. Dean Krasinski of Old Dominion University's Electrical and Computer Engineering Department. As Ms. Graves said, I found Dr. Krasinski through an Ocean Lakes Academy student from the class of 2013. While at ODU, I attempted to identify people based upon their brainwaves, a process called electroencephalogram biometrics. Essentially, I use data collected from a cap like the one pictured here to look for individuality in brain signals. In order to fully explain EEG biometrics, I will first give a brief explanation of biometrics in general. Some of you might recognize this person. But how? How do you know this image is of our president? The answer to these questions is biometrics. Biometrics involves directly measuring someone's traits to establish or verify their identity. When you look at someone, or even a photo of them in my PowerPoint, your brain instantly classifies them based on several traits. These might be hair color, eye color, or any other physical characteristic, but the important thing is that you can decide who someone is based upon these traits. Traits are central to biometrics. Biometric systems are more prolific than you might realize. If you own an iPhone 5S, you have a fingerprint scanner in the home button. This is a biometric system. It reads your traits directly, your fingerprint, and decides if you are the owner of the phone. This computer-based decision process is a classification. When you view a picture of Barack Obama and recognize his identity, you are performing a classification in your brain. Most digital biometric systems have a single trait they observe, a fingerprint in the case of the iPhone. This trait is also referred to as the modality, or the method of classification. Some common modalities are shown here. As I previously stated, my project investigated the viability of EEG biometrics, biometric systems with brainwaves as the modality. But how does the modality relate to a biometric system? And how does it function? Biometric systems generally have two forms, verification and identification systems. Verification systems work by first creating a definition of each user for future reference. This is a sampling of a modality called features, which are stored in a database on the far right of this diagram. For your iPhone's fingerprint scanner, features are the pattern of ridges on your fingerprint. Besides creating this definition, a biometric system must also define an acceptable level of intra-class variation, the differences that might arise between multiple trials of a single person. Each measurement of a single person's fingerprint will be slightly different, so the system needs to understand where acceptable differences end and other users begin. Finally, the system needs to be ready to accept new measurements, compare them to the stored profile, and classify the data. For verification systems, this classification simply tells the user if they are admitted or not, rejecting them if they do not match the profile they claim. This final decision is shown at the bottom of the diagram as true or false. There also exists another form of biometrics, identification systems. If you've ever watched crime shows like CSI that use DNA to identify criminals, you might be familiar with this form of biometrics. Identification systems compare new measurements to multiple profiles, not just one. This is a one-to-many comparison. The identification system will decide if the input data is linked to anyone in the database, also using acceptable levels of differences in features to classify someone. However, this type of system can classify the novel data as anyone in the database, or even reject them completely by responding that the person is not in the database at all. In my research, a hybrid approach was used because EEG biometrics are still currently in development, so acceptable levels of differences between subjects are not well defined. My hybrid approach allows each user to declare their identity, as is done in verification systems, before the computer conducts a one-to-many comparison, as is done in identification systems. This approach was advantageous because it allowed me to evaluate brainwaves as modality without placing too much emphasis on the practicality of the system I was creating. But you might be wondering why biometrics is necessary to develop in the first place. Your student IDs and passwords here at Ocean Lakes work fine to verify your identity, but this is not a biometric system. Why are biometrics necessary to study if password systems work fine? The answer is that password systems are not appropriate for various applications. In high security situations, especially digital government and banking systems, biometric traits are important because they cannot be stolen, at least to a greater degree than passwords. However, there is still a chance for the traits measured by more public biometric systems, like fingerprint scanners, to be stolen. Fingerprints are left on everything we touch, and a malicious user could feasibly steal fingerprints with enough motivation. For this reason, my project focused on an even less public of a trait than fingerprints, brainwaves. The research hypothesis I used was that if Euclidean distance measurements were used for verification, then the classification accuracy would improve as compared to random chance. The null hypothesis was that the method of evaluation used would have no impact on classification accuracy. For my research, I defined these brainwaves as the activity of the occipital lobe while the subjects viewed a flashing light at a steadily increasing or decreasing rate, 5 to 20 or 20 to 5 hertz, over 30 seconds. The electrode locations I used are shown on the screen. This is a top-down diagram of electrode placement on the subject's head. The numbers are simply the numbers of the electrodes 1 through 16. The electrodes are used to record the brainwaves and translate them from analog, human biological signals to digital, understood by a computer. In order to establish a machine learning approach, I separated the trials recorded into training and testing data, the former to create a profile in the database, and the latter to test the accuracy of the machine by performing classifications. However, the raw EEG potentials recorded were not appropriate for biometrics and are notoriously difficult to analyze. Therefore, my modality was not actually EEG brainwaves, but the component sinusoids that define a brainwave in the frequency domain. These component sinusoids were calculated through Fourier transform. Fourier transform uses the notion that complex waveforms can be written as the addition of simpler parts to translate functions between the time domain and frequency domain. It has its roots in Euler's formula, which states sinusoids can be written in a basic exponential format. A Fourier transform checks for the relative presence of different frequencies within a complex waveform. For instance, we can calculate the component frequencies of the oscillating function on the board. As time, graph on the x-axis goes on, the function jumps back and forth above and below the axis. However, this is not a simple sinusoid. There are many sinusoids that together sum to create this shape. Fourier transform can be used to calculate these component frequencies. This function will output a value that represents the presence of a frequency zeta in f of x. If an exponential term from Euler's formula is applied to the original function, the resulting expression will have limits to infinity and negative infinity of 0, allowing for the integral over the infinite time domain to be calculated. This calibrated limiter is specific to the frequency you're looking for because the zeta appears in the exponent. The integral is used to calculate the total accumulation of the frequency over the entire domain. This accumulation of the selected frequency yields the frequency's relative presence in the original function, meaning that once this entire process is completed, we know how prevalent a single frequency zeta is in the original function. This is an example of a limited function upon which Fourier transform performs integration. At this point, we have applied a limiter, but we have not integrated. The spikes are the areas of the domain over which the selected frequency is most present. This is where the limiter is in phase with the graph. There are two graphs here, one for the real and one for the imaginary dimension. Therefore, all Fourier coefficients are complex numbers, but in my research, the imaginary dimension was not relevant. The light blue simple sinusoids are the components, all calculated in the manner I just outlined. Their sum yields the original red function. This next picture relates the time domain, graphed in red, to the frequency domain, graphed in blue. The complete Fourier transform for the original function is shown for positive real frequencies on the right-hand side of this picture. The spikes are essentially a side view of the sinusoids that together sum to create the red function, allowing their different amplitudes to be graphed against their frequency. Once more, here's the final transform graphed in the frequency domain. This picture is the same function as the red function, it's just a different type of domain. This is frequency versus time domain. In my research, a discrete time Fourier transform was used since EEG signals are digitized from brainwaves. A discrete time Fourier transform works because of the same theorems and formulas, but is calculated on discrete data. Once Fourier transforms were used to develop the features necessary for biometrics, these spikes, the classifications could occur. Classifications in this research utilize Euclidean distance measurements in high-dimensional space to compute the distance between a subject and the different profiles of the database, matching subjects that are closest in the virtual database space. Many of you might be familiar with the distance formula, which computes the distance between 2 points on our Cartesian plane. This formula is essentially a truncated version of Euclidean distance made to function in 2 dimensions. There's actually no limit on how many dimensions that Euclidean distance can be used upon, and in this research, the number of dimensions ranged from 40 to 640. Euclidean distance was harnessed to classify subjects via the logic that if a subject was closest to a particular profile in the database, then they must be that subject. The distances were calculated with each component sinusoid from each channel as a dimension. In the formula shown, each squared binomial term is a dimension. These were the features or specific components in the modality used in classification. So recall my machine learning approach. The P set represents the training data, and the Q set represents the testing data. Therefore, Euclidean distance is a function of the difference between these two sets. So as two sets, maybe subject 1 and profile 1 in the database, are more similar, the distance will decrease. I performed several classifications, one for each electrode and 2 comprehensive measurements using multiple electrodes. The results of these are shown now. The terms channel and electrode are used interchangeably here, and the numbers 1 through 16 correspond to the top-down image I showed earlier. The 2 comprehensive measurements differed in the number of features and therefore the number of dimensions used. Dimensionality reduction was achieved by averaging 4 Hz segments into a single feature and selecting 9 channels as a representative sample of the scalp. The classification accuracy was retained through this process, resulting in a successful dimensionality reduction. The reduced dimensionality comprehensive measurement was the least likely to be the result of random chance. This is the second row with 16 out of 21 subjects verified. Overall, my p-values range from 3.07 times 10 to the negative 6th to 1.66 times 10 to the negative 19th. The p-values in the far right column were calculated as the probability that any number of subjects could be verified correctly via only noise. This is based on a binomial distribution, which combines the probability mass function with the combination formula. The left term is the number of ways to arrange k correct verifications, and the right term is the probability of actually achieving k correct verifications. The 2 terms multiplied yield the probability of any number of verifications, in my case 0 through 21. This is an example binomial distribution for a fair coin. The odds are 50-50, so out of 50 flips, 25 heads is the most likely outcome, designated by the highest peak. Each peak was calculated with the formula I just showed. This theory will later be applied to my study. As the probability shifts lower, the distribution shifts as well. The rightmost distribution is the same as the slide before, 50-50. Each subsequent distribution is 10% less likely for the desired outcome, like an increasingly unfair coin. My research used 21 subjects, so the probability of a subject being verified randomly was 1 over 21. The distribution I used is shown, with arrows pointing to the highest accuracy that my research yielded, 16 out of 21 subjects. By understanding the probability that my results could arise from statistical noise, a judgment of their significance can be made. The odds on the board are that very probability. This will never be reduced to 0, but if it is acceptably small, then conclusions can be made regarding my hypothesis. The probability of 16 subjects being verified from only noise was approximately 1 in 10 quintillion. This is an unimaginably small chance. For scale, a comparable ratio exists between your fingernail and the entire surface area of the planet Earth. Based on the fact that my results were likely not because of random chance, conclusions can be made regarding my hypothesis. My research supported the research hypothesis that if Euclidean distance measurements were used for verification, then the classification accuracy would improve as compared to random chance. This did not support the null hypothesis, that the method of evaluation used would have no impact on classification accuracy. This is generally in agreement with current studies on EEG biometrics. Additionally, according to the varied results in channel performance, some regions of the occipital lobe are more suitable for biometrics than others, as shown. Simply, this research demonstrates that brainwaves could realistically be developed into a biometric modality. Although I effectively supported brainwaves as a modality, there is a wide range of potential continuations on my research. My research was heavy in statistics and the development of a suitable procedure for data analysis. As a result, not much emphasis can be placed on the data collection side, as I borrowed data from an ODU graduate student that Dr. Krasinski recommended for this analysis. In future research, I would attempt to investigate differing stimuli and the different brain regions associated with them. Furthermore, I would like to investigate a resting patient with no stimulus to see how those results compared to the results of this study. My research experience was overwhelmingly positive. I spent the summer in an intellectual environment researching a genuinely interesting topic while learning more than I could have ever imagined. The only drawbacks were the large amount of background research required to jump into a field this complex and the short time frame over which I completed my study, limiting my options in data collection. Despite these drawbacks, I'm appreciative of the opportunity to work in such an amazing field under such an intelligent ODU staff, which brings me to my final point. I have several people to thank for this experience. First and foremost, my advisors at OL and ODU, Mrs. Green, Mrs. Graves, and Dr. Krasinski. Additionally, I would like to thank my parents, especially my father, who's been stationed in DC while I've completed the magnet program. I'd also like to thank the mathematical role models who have inspired and amazes me. Finally, I would like to thank all of you for attending my presentation. And now, any questions? Sarah.
I think you mentioned something about dimensionality reduction. Can you talk a little bit more about that?
All right, so dimensionality reduction was the process I had between my two comprehensive measurements. So the first one had 640 features used. So I used all 40 from 1 to 40 hertz from every channel, 16 channels, and that multiplied yields 640. So I had 640 features in 640 dimensions in the first comprehensive measurement. However, there's something that is loosely referred to as the curse of dimensionality, which is when you increase the dimensionality of a space, you're also increasing the baseline, essentially, distance between two points in that plane. So more dimensions essentially means that the ratio of signal to noise is changing in a way that you don't want it to. You want your signal to be a lot more greater than noise, and increasing dimensionality is increasing your noise because there's a general level of distance that comes between subjects in a high-dimensional space. Yes, Ms. Gray?
What do you mean by the term noise?
So essentially, noise is the part of your data that you're really not interested in. If I was looking at a brainwave or looking at a set of data and they were all, let's say, 100, and the variance, the actual signal, the difference between them was like some small fraction, like 0.01. You have a lot of noise, that like 100 ratio to your signal. So what you want is you want to have a signal that's easily measurable. So this is why you had to reduce the dimensionality of the space. So representatively, I picked 9 channels on that diagram that was a symmetric and representative sample of the head in order to have measurements that were reduced in noise ratio. Yes?
Why are most electrodes of the EEG located in the posterior region of the brain?
Okay, that's a great question. So he's asking why those electrodes were the ones I used. So the subject was viewing a flashing light. In the occipital lobe, which is that posterior region of the brain, this is where visual information is processed. So we were looking generally in that region because we were looking at how the occipital lobe would respond, because generally the other regions of the brain would not respond much to a visual stimulus. Yes?
Kevin Nelson, what classes and experiences at Ocean Lakes High School helped you with this project?
That's a tough question, because really it was both all my classes and none of my classes. Because the most important thing I learned at Ocean Lakes was how to learn, and all of my classes help with that. But when I'm in this crazy environment at ODU researching graduate level stuff, there's not much experience I have from Ocean Lakes. So how to learn was the most important thing that I got out of all my classes at Ocean Lakes. But at some point, I had to do some of the research on my own. I had to be willing to read scientific papers and stuff like that. There was not much prior experience at Ocean Lakes. Yes?
What were some interesting things that happened to you while you were doing your research project?
Interesting things? As far as, it was going to the ODU lab and sitting in front of my computer and occasionally asking for advice, so there wasn't, it wasn't a whole exciting experience as far as that goes, but the actual content was the most exciting and interesting part of it. Yes, Ms. Graves?
So you yourself never hooked any of your subjects up to the EEG machine. You're using someone else's data?
Yeah, so there's a team of graduate students that work under Dr. Krasinski. So there was one in particular where he said that visually evoked potentials is one of the areas that EEG biometrics are typically studied in the past few years. So I was looking for a visual stimulus specifically, and I got it from a graduate student there. So I never actually saw any kind of patients or subjects or anything. I was just looking at the data I had, which is why it ended up actually being not completely optimized for biometrics, although I was still able to support biometrics.